pharmacists are routinely confronted with the unhealthy behaviors of people with substance use disorder. We do not like to see ourselves enabling these addictions, and we have some very serious legal responsibilities to governing bodies and upholding process and law. While the laws we are charged to uphold are frequently unbendable, the manner in which we interact with the human is. The purpose of this program is to examine aspects of addiction and neuroscience with brief discussions of a few substances of abuse. Many of the elements we'll cover are in fact highly complex. Some could require hours and hours to thoroughly discuss. However, during this hour-long program, we're simply taking glimpses into some current understandings of neuroscience and addiction for the purpose of expanding our knowledge and improving our care. Our scientific bodies have imperfect knowledge of addiction, and there is still much to learn about our brains and the deadly disease of addiction. Addiction science is rapidly evolving, and pharmacists can be key treatment team members. But what about all those off-putting behaviors of the addict? Our rational minds just can't wrap around it. We see the addict repeating annoying and harmful behaviors over and over, despite awful consequences. It just makes no sense. Before we get going, let me tell you just a little bit about myself. Through a confluence of serendipity and saying yes to an interesting opportunity way back in 1988, post-graduation, I found myself in psychiatric pharmacy. I was assigned to the team that treated many of what we then called the dual diagnosis clients. In 1994, a colleague and I at Tennessee's Association of Mental Health Centers annual meeting gave a presentation titled The Odd Couple. It was a well-attended presentation that was a case study on how we were approaching the care of a dually diagnosed client. The client was male with diagnoses of bipolar disorder with psychotic features and alcohol use disorder, amongst other things. Additionally, he had a couple of serious suicidal attempts resulting from both his hallucinations and the self-loathing he experienced from his alcohol abuse. I can tell you we did get a few things right, we got a lot of things wrong. Fortunately for us all, our understanding and treatment of these humans with very complex diseases has come a long way since then. In addition to my professional experience, like many of us, I have family and friends who have suffered from the disease of addiction. So, while addiction is not one of my struggles, it could be said I've been riding professional and personal shotgun with it most of my adult life. Psychiatric pharmacy and neuroscience hold such a charisma for me. Last summer, a friend of mine, who is as kind, gentle, and modest a person as one can be, had a small thalamic stroke. Until that moment, he had handled all his own affairs and was a savvy manager of his own funds. Once recovered, he appeared to be very much himself, but his concept of numbers was gone. He could write a $4,000 check and lose it with little more concern than if he'd lost 40. Through this change, no one, not his family nor his friends, judged him irresponsible or bad. It was recognized by all that he had a cognitive change that resulted in behaviors that were no longer in alignment with his value system. His support system simply adjusted to this without judgment. This event and how those who loved him responded and adjusted was quite an epiphany. Now, on to the subject of the program. In 2019, the American Society of Addiction Medicine updated their definition of addiction to that of it being a treatable chronic medical disease involving complex interactions among brain circuits, genetics, the environment, and an individual's life experiences. People with addiction use substances or engage in behaviors that become compulsive and often continue despite harmful consequences. Prevention efforts and treatment approaches for addiction are generally as successful as those for other chronic diseases. Viktor Frankl, born 1905, was an Austrian neurologist and psychiatrist, as well as a Holocaust survivor. He described between stimulus and response, there is a space. In that space is our power to choose our response. In our response lies our growth and our freedom. I say to you, as I tell my own daughters, That space is where real magic happens. Frankl's charge of choosing our response especially applies to healthcare providers. Non-addiction, specialty-trained healthcare members providing care to any of the addicted population can be confounded by the behaviors witnessed. Our humanness, our biology, drives us to reject these people we mistakenly identify as aberrant. We witness these unhealthy, destructive behaviors and too frequently conclude the person has made conscious choices that have led them to this moment. Joshua Green, author of Moral Tribes, Emotion, Reason, and the Gap Between Us and Them, writes of our brains having evolved for tribal life, for getting along with those like us, and the fighting of the thems. How could we grow if we incorporate neuroscience, often much advanced from our days of formal education? What growth could be allowed into our own lives, as well as our professional potential? Yuval Noah Harari, author of 21 Lessons for the 21st Century, a book I highly recommend, writes, "It turns out that our choices of everything from food to mates result not from some mysterious free will, but rather from billions of neurons calculating probabilities within a split second." He goes on to describe how all this is taking place just out of range of our consciousness. That these calculations are happening mostly outside our consciousness fosters our frequent misapprehensions about the nature of choice and volition. Could addiction be better described as a disease of volition? Volition being the power of using one's will. How does volition or choice work? How does the brain make choices? And can the way a brain makes choices become broken? So let's talk about how our brain makes some decisions and shatter the myth of choice. 3 aspects of addiction we are beginning to understand are: first, there is a disorder of pleasure, meaning how a brain perceives pleasure and the consequences of a broken pleasure sense. A blind person cannot perceive light, and an addict cannot perceive pleasure correctly, and that undermines their ability to choose. Second, there is a disorder of choice. Our reward system has to function properly in order to make choices informed by past consequences, predict future consequences, and to align with our values. Third, it can be influenced by stress. The dopamine pathways are sensitive to stress, especially early childhood stress, and even inherited stress. Substance misuse, such as intoxication, is also a kind of stress on brains and can be sufficiently stressful enough to break the dopamine system. In the following section, we'll look at our brain's cortex, which plays key roles in attention, perception, awareness, thought, memory, executive decision-making, and consciousness. It is well documented that with extended substance abuse, the cortex loses function and volume. With that loss, it is reasonable to expect some consequences. Let's examine 3 cortex regions that, if impacted by this decreasing function and volume, could help us gain insight into addict behaviors. 1. The orbital frontal cortex at the front of the brain and located just above each eyeball assists us with assigning values. If an idea, item, or person enters my life, I use my orbital frontal cortex to assign value. Conversely, if someone has a defect within this region and you were to place a VW and a BMW in front of that person, she or he could have difficulty in assigning a higher value to the BMW. Our ability to assign appropriate valuations is an important component of volition. It is an essential mechanism in our ability to make a choice. People who develop orbitofrontal defect will struggle with valuation, which can contribute to their losing their jobs, their marriages, and so forth. 2. The anterior cingulate cortex assists in higher level functions of performance monitoring and error detection. It works along with the orbitofrontal cortex in reward anticipation. This part of the brain helps us see our behaviors through another's eyes. It allows for self-monitoring based on feedback. When this ability is diminished, the affected person, who may otherwise be kind and considerate, to potentially be cruel. Ask any mother of a child with an opioid use disorder, and she'll be able to provide countless examples. 3. The insular cortex is a small structure located in the folds of the cortex and, amongst other things, appears to be part of our brain that attaches our consciousness to our body. It links sensory input from our body to our brain and is involved in cravings and our ability to direct attention to cognitive control and away from visceral cues, or the sensory input, meaning modulating the insular cortex may help restore cognitive control over cravings. One can see that dysfunction in one or more of these areas could lead to poor decision-making. There is an impairment in the ability to make healthy decisions, and to make matters worse, the feedback loop can be broken so the person is blind to it. These are some examples of the disorder of choice aspect in addiction. This is a good time to revisit the unfortunate case of Phineas Gage. Mr. Gage was born in 1823 and is known for surviving a 3-foot 7-inch, 13.5-pound bar being accidentally blown through his skull and in the process taking a bit of his brain with it. Prior to the accident, Mr. Gage was well-liked and professionally successful. After the shock of surviving such an accident, and with time regaining his physical strength, was able to return to work. He seems to have shown no motor, speech, or memory impairment. However, he is described as becoming fitful, irreverent, and grossly profane, showing little deference for his fellows. The misfortune of Mr. Gage is an early documented example of acute changes of behavior and choice, resulting from a pretty tangible alteration to his brain. While this story is sad indeed, it illustrates the complexity of our brain, and to this day, science is laboring to learn of its vast and elusive mysteries. Next, we'll take a look at the limbic system and begin exploring some of the aspects of the disorder of pleasure. We see ourselves as human first, but in many areas of our brain, we are simply biological creatures that need to be kept alive long enough to reproduce. In the next section, we're going to discuss 2 dopamine pathways of our reward system. First up is the Mesolimbic Dopamine Pathway, also known as the reward pathway. It begins with the ventral tegmental area located in the brainstem. This area releases dopamine when exposed to anything pleasurable. The mesolimbic dopamine pathway connects the ventral tegmental area to the nucleus accumbens, which is located in the ventral striatum and is part of the brain associated with reward. When we experience something pleasurable, the dopamine neurons in the ventral tegmental area are activated, and these neurons project into the nucleus accumbens via the mesolimbic dopamine pathway, directing a rise of dopamine in the nucleus accumbens. This reward pathway, utilizing dopamine, creates a sense of anticipation for a stimulus. It is the way our brains evolved to assign importance to environmental stimuli, such as food, sex, and shelter. Dopamine helped our forebears take note of things required for our survival by recognizing things that are good, learn them, and be able to predict them. Dopamine also takes note of when something is better than anticipated and drives me to seek it out again. When something produces a larger dopamine release than anticipated, the brain then moves that item up the hierarchy of survival needs. The system is designed to sort out and assign hierarchy to survival salient stimuli. Our understanding today is every addiction is associated with the activation of the mesolimbic dopamine pathway. The larger the dopamine released through this pathway, the more importance the brain assigns to that stimulus. The drug becomes increasingly important. This is the hijacking of the brain. Spoiler alert, these things all create dopamine spikes along the dopamine pathway that predictably result in downregulation of dopamine receptors due to homeostasis. Decreases in dopamine receptor density are going to make the ordinary world less and less interesting, and is also going to increase our vulnerability of cross-addiction. We'll cover homeostasis and downregulation in some detail later. Through addictive substance exposure, our behavior-brain balance is shifting to overvaluing the pleasurable, while simultaneously our cortex is losing its ability to assign healthy values and having intact insight into the consequences of our choices. The second dopamine pathway we'll look at is the mesocortical pathway, which is also considered part of the reward pathway. The mesocortical pathway also begins in the ventral tegmental area, but travels to the frontal lobes in the cortex. The mesocortical pathway is also activated during pleasurable experiences. Let's take a quick look at glutamate, which is our predominant excitatory neurotransmitter. Dopamine release results in a release of glutamate that is proportional to the dopamine release. With dopamine spikes from addictive stimuli, you will also get glutamate spikes that can create powerful memories. These memories can include seeing the needle, the people one was with, even smells. These are examples of triggers. These glutamate spikes create powerful memories that can be triggered out of the blue after days, months, and years from the last use. We are going to touch on the role of associations, triggers, and opponent process later as we discuss homeostasis. Genetic variability is well-established science, and while our genes do not cause addiction, they contribute to how we experience substances. A couple examples of genetic variations contributing to differing substance experience are low versus high responders to the effects of alcohol. If one is a low responder to alcohol, it requires more alcohol to be consumed to achieve the desired intoxication effect, resulting in higher alcohol levels being on board and subjecting that brain to higher stress from that higher alcohol level. As a result, that individual is likely to consume larger amounts than their contemporaries, increasing the stress on their brains and their risk of developing alcohol use disorder down the road. Or an individual could be a super responder to opioids, meaning even if the person is legitimately using an opioid pain medication for appropriate purposes as prescribed, their brain may respond with a surge of dopamine, and that person's experiences euphoria. The resulting euphoria will lend itself to more frequent use, placing that individual at higher risk of developing opioid use disorder with time and repeated exposure. We are all born to this life with a multitude of genetic strengths and weaknesses. ASAM, the American Society of Addiction Medicine, states that genetics account for about half of one's likelihood to develop an addiction disorder. Next, we'll look at epigenetics and a few types of stress that relate to addiction. On top of our genetics is epigenetics. I'm riveted by the very idea of epigenetics, which is the study of organism changes caused by modification of gene expression rather than the genetic code itself. There's not a change in the DNA sequence, but instead how the cells read the genes. It seems to open the door for some information that is being experienced today by an organism to have transgenerational effects. From an evolutionary lens, that's pretty interesting. Epigenetics is a fairly new science, and any role it may or may not have on addiction is virtually unknown at this time. But understanding it as a data-supported science is an important consideration. It is fairly easy to see how the experiences of a pregnant woman affect the developing reproductive cells within that unborn child. Thus, some experiences of the pregnant woman can have impact on that future grandchild, but epigenetics goes beyond that. By the way, through epigenetics, it's not just the experiences of females that have transgenerational influences, but also the males. I find that a nice change. A famous epigenetic study is the Överkalix study, for which there is abundant published work. Överkalix is a small, isolated municipality in northeastern Sweden that is routinely cut off during severe winters. Studies have been conducted utilizing historical records, including the records of harvest and food prices. From this, we've learned there are remarkable transgenerational findings that are linked to environmental factors. As but one example, it was realized that if a just prepubescent boy is exposed to food scarcity, perhaps as a result of a crop failure. Those boys' future grandsons will have a decreased rate of cardiovascular disease, seemingly due to a reduction of the risk of developing diabetes mellitus. Their grandsons live longer than those whose grandfathers had experienced plentiful food during prepubescence. As a note, other studies suggest cancer mortality was a contributing factor rather than diabetes and cardiovascular mortality. An intriguing aspect of these studies is the conjecture that an epigenetics pathway may open up just before puberty during the slow growth period, and this being but one of the windows for germline reprogramming. What there is to be learned about epigenetics may rock our scientific worlds. Now, moving on to the role of stress. We touched on consequences of brain stress of intoxication earlier, but there are others. The hypothalamic pituitary adrenal axis is a complex set of direct influences and feedback interactions, releasing cortisol to help us address stress. Under normal conditions, once the source of stress has been addressed, there is a negative feedback to the hypothalamus and anterior pituitary informing them all has been dealt with, all is well. However, in toxic stress, including stress from intoxication or violence or poverty, the stress system is continuously in overdrive. This chronic exposure to stress can result in a downregulation of the dopamine receptors. What follows then is a paucity of these receptors, which can lead to a condition called anhedonia. Anhedonia can be described as pleasure blindness. Now the person may find normal pleasurable experiences may not be seen, but high dopamine releasing events can. Thus, you have a snowballing effect. Spiking dopamine from high dopamine releasing stimuli is increasingly what is required to push back against stress. And over time, the substance is used to address all stress. While we're on the subject of stress, let's touch on adverse childhood stress and our increasing appreciation of its consequences. This is the briefest of descriptions because this subject alone could require hours and hours to cover, but it is imperative that we understand its role in wellness and disease. The Division of Violence Prevention at the CDC, in partnership with HMO Kaiser Permanente, conducted the landmark Adverse Childhood Experiences Study, also known as ACEs, from 1995 to 1997. Since then, numerous other studies have been conducted with similar results. 10 events of concern were revealed through ACEs. As one's ACE score increases, so does their risk of a wide range of outcomes. Findings include ACEs are common and have a dose response with many health problems. Over time, it was discovered that an individual's cumulative ACE score had a strong graded relationship to numerous health, social, and behavioral problems through one's lifespan, including substance use disorders. One of the study's many findings is that some survivors of childhood trauma were, amongst other things, as much as 4,600% more likely to become IV drug users. Yes, you heard me correctly, and let's pause over that for just a moment. From the Origins of Addiction, Evidence of the Adverse Childhood Experiences Study, written by Dr. Vincent Felitti, I quote, "For instance, a male child with an ACE score of 6 when compared to a male child with an ACE score of 0 has a 46-fold or 4,600% increase in the likelihood of becoming an injection drug user sometime later in life. It is increasingly held that without factoring in the patient's childhood experiences, the healthcare provider is missing a significant piece of the puzzle in the treatment of many diseases." We've referred to homeostasis a couple of times. Now let's take a look at what it is and its relation to addiction. Biologists hold that our mental and emotional world is governed by biochemical mechanisms shaped by millions of years of evolution. Our subjective sense of well-being is not determined by external parameters, but rather by a complex system of nerves, neurons, synapses, and various biochemical substances. All these pieces work in concert to keep us at homeostasis. As an example, the human species depends upon sex for survival. Through the reward system, evolution provides pleasant feelings as reward for having sex. If sex were not accompanied by pleasure, few would bother. At the same time, evolution made sure these pleasant feelings are transient and quickly fade. If orgasms were to last forever, we'd die of hunger for lack of food. To keep us alive, our bodies and brains drive us to seek pleasure, but also to return us to set point. Some compare our biochemistry to an HVAC system. Outside it may be a snowstorm or a heat wave, but the system is designed to keep the internal temperature constant. Within the range of ordinary experiences, some events temporarily cause small temperature variations, but the system will work to reestablish the temperature to its set point. This is homeostasis, and it is for this reason the effects of drug use are always temporary. Our biochemistry will assert its set point through all manner of adaptations and compensations. This predictable biological response is the bane of anyone who is trying to maintain elevated dopamine levels through the use of addictive substances. The efforts of our biological imperative to return to set point will not subside, confounding the urges and drives of the user. Homeostasis will assert itself. Evolution demands it to be so. Neurobiologists refer to our brain's capacity for modification as plasticity, which is our brain's ability to modify itself to adapt to environmental input. The learning associated with addiction begins with the very first exposure to a drug. Tachyphylaxis refers to the immediate adaptive compensatory changes taking place as an altering substance reaches the brain. This is a result of our biochemistry's effort to return to set point or homeostasis. These changes can be relatively minor and transient, as in the case of the occasional drinker. When one drinks alcohol, the rising alcohol level increasingly affects the function of the brain, and the brain immediately begins to rebalance the activity dampening effects of the alcohol by becoming more excited than normal. We notice this adaptation the next morning when lights seem too bright, sounds too loud. Fortunately, by the afternoon, homeostasis has continued to seek balance and the hangover is over. However, when alcohol ingestion is being repeated with chronic and increasing amounts, the effects become more pronounced and lasting. As the brain is adapting to the abused drug, these adaptations result in the drug becoming less effective. The user must take more and more of the substance to produce the same experience. This is drug tolerance. Thus, the user has begun the ever-increasingly futile journey of trying to replicate that first high, resulting in more and more brain adaptation. Eventually, the person's brain is so well adapted to the effects of the drug, all the user can hope for is a transient relief of chronic despair and misery. Dr. Judith Grisel, author of Never Enough, The Neuroscience and Experience of Addiction, writes of Solomon and Cobert describing the opponent process theory of motivation and emotion as pairs of opposites. With repeated stimulation, the opposing emotion becomes stronger and weakens the experience of the primary emotion. The user gains positive feelings through use. This is then countered or opposed by the drug opposite effect. As in our earlier example, the increase in excitation of our brain to counteract the activity dampening effects of alcohol ingestion result in the negative feelings the next morning. This idea led to the name of her book, Never Enough. Our biology, our evolution requires our biochemistry to return to homeostasis no matter the wishes or desires of the user. When our brain is first exposed to a stimulus, the initial process is maximal and unmitigated by any compensation. With each use thereafter, the opponent process is strengthened. Dr. Grisel explains that our brains are so evolved to counteract disruptions to our homeostasis, and with enough exposure to the stimulus, we'll learn to anticipate these disruptions, rather than wait for the disruption to occur, and will begin the opponent process even before the initial process. She explains this happens even at the casual level. As an example, say I like to share a particular cocktail with my husband at home every Friday night, and I've been doing this for years. That cocktail over time has a diminished effect due to its repetition of the same event. The cues, that cocktail, Friday night, my husband, trigger my opponent process even before I take my first sip. If I were to consume the very same cocktail on a different day, time, place, and company, the result would be a greater initial process. I'd have a greater response to that same drink. With substantially increased frequency, duration, and amount, a body will elicit powerful opponent process experienced as symptoms of withdrawal and craving by simply being exposed to the associations the brain has learned to indicate substance use is about to occur. It is not unusual for substance use treatment practices following detox and sufficient stability to, in measured amounts, reintroduce those associations or triggers, such as a payday, experiencing frustration, walking into a Quick Mart. Early on, these events will likely result in the opponent process, such as changes in pulse and respiration. The good news is, with these repeated exposures and no drug being delivered, the opponent process will diminish and with enough time eventually disappear. Next up, we'll look at a few substances of abuse. In the next section, we're going to take a look at a few addictive substances: alcohol, opioids, THC, amphetamines, and psychedelics. But before we begin discussing these 5 agents, let's remember at its simplest, an addictive substance is a stimulus that causes a spike of dopamine through the reward pathway, resulting in various compensatory adaptations, including downregulation of dopamine receptors. Tobacco smoking is just such an addiction. Per the CDC 2012 Compendium, as many as 25% of adults and 17% of youth smoke. 480,000 deaths were caused by this legal substance each year. As a point of perspective, just imagine if these deaths that we barely register were the result of a terror attack or a new disease such as COVID. And if that is not enough, through the downregulation of the smoker's dopamine receptors, the smoker is also more vulnerable to other addictions. The CDC asserts excessive alcohol use is responsible for 88,000 deaths per year in the U.S. In 2018, the World Health Organization reported that worldwide 3 million people died from the harmful use of alcohol, and goes on to report harmful use of alcohol is a causal effect in more than 200 disease and injury conditions. All these folks are dying quietly around us every day with barely a notice. Since alcohol has been consumed by humans for thousands of years, one could say we've evolved with it. Considering the time we've been consuming alcohol and it being a remarkably simple molecule, one would think we understood the effects pretty well. But quite the opposite. We understand relatively little of its mechanisms of action. The smallness of alcohol molecule makes its mechanism of actions difficult to pin down, unlike THC, opioid, and amphetamine molecules, which are relatively large, resulting in specific sites of action. Alcohol is small and can less specifically fit into many more receptors. As a result, we have less understanding of how alcohol makes us feel the way it does. Some of the sites of action of alcohol identified include: alcohol's foremost action appears to be its facilitation of GABA neurotransmission. GABA is a primary inhibitory neurotransmitter in our brain. With GABA neurotransmission activated, neural activity slows down. With a moderate dose, one feels relaxed, and if consumption continues, they'll feel sleepy and then pass out. Alcohol reduces activity at the glutamate receptors. Glutamate is our brain's primary excitatory neurotransmitter. Activity here impairs the formation of memories, and with higher doses resulting in blackout. Note, it is the opponent process of GABA activation and glutamate inhibition that places acute alcohol abstinence for those with alcohol use disorder at risk for seizures and death. If you'll recall our biological homeostasis imperative, with time and continued alcohol misuse, the resulting repeated GABA neural activation and glutamate inhibition adaptations are occurring. The net result of these adaptations is decreased GABA and increased glutamate. If alcohol is then acutely withdrawn, the adaptation is revealed, and one is left with excessive and dangerous levels of neural activity, and hence seizure risk with resulting risk of death. It is also worth noting that in part because GABA and glutamate having activity sites throughout our brain, not just in a few pathways here and there, that helps us understand the global impact of alcohol. Its effects are manifested through a generalized slowing of neural activity, resulting in impaired motor, cognition, emotion, and memory. As with all addictive substances, there is the activation of the Mesolimbic pathway, resulting in pleasant anticipatory feelings. In alcohol's case, it is thought the activation occurs by way of the opioid receptor being activated by our own endogenous opioids. It is held that at low to moderate levels of alcohol, there are increases in the synthesis of beta endorphins, which then contribute to alcohol euphoria through action in the mesolimbic system. It is this side of action that naltrexone interrupts. As an important note, naltrexone can be an effective treatment tool for some with alcohol use disorder as they struggle with sobriety. Let's pause here and consider this mechanism of action, because we find we've circled back to genetic variation having impact on the risk of developing alcohol use disorder. If one were to have an enhanced ability to synthesize beta endorphins through some genetic variability, that would lead one to experience excessive euphoria or well-being with alcohol consumption. It is easy to see that such an individual might chronically consume larger and larger quantities of alcohol, with the resulting risk of developing alcohol use disorder with time and exposure. Alcohol is also involved in a mishmash of other actions, and how these actions make us feel is less well understood. Some examples are: alcohol slows neural activity through actions on calcium channels, calcium being essential for neurotransmitters being released into the neural synapses, which in turn decrease neural effective communications. At higher levels, alcohol begins to affect myelination quality of the nerves, further decreasing neural activity. And it also appears to interact with serotonin and acetylcholine. As you can surmise, while most drugs have a fairly specific site of action with specific effects, this smorgasbord of actions makes sorting out the mechanisms of drunkenness a bit challenging. Opioids and opiate terminology can be a little confusing. Opiate is an older term and used to describe substances derived from opium. Now, opioids designate all substances, natural and synthetic, that bind to the opioid receptors in our brains. Opiates are compounds naturally found in the opium plant, such as morphine and codeine. Other opioids can be synthetic and semi-synthetic, such as hydrocodone and fentanyl. Additionally, there are opioid antagonists like naltrexone and endogenous opioids known as endorphins. Our brains manufacture numerous and varied natural opioids. Their sheer number and wide distribution portends that from an evolutionary perspective, they play an important role in our survival. It is held that sensing pain is essential to our survival. When you compare it to our other senses, it is easy to see it must be especially critical. Blind or deaf people can live virtually normal lifespans, whereas persons with congenital insensitivity to pain are rare, and they typically die young. In fact, congenital insensitivity to pain is an extremely rare disorder, and one of the reasons scientists believe it is so rare is few persons with the disorder reach adulthood. We can conclude from the evolutionary and biological perspective, experiencing pain is essential to survival, which explains our body's robust compensatory actions to return an opioid user to homeostasis. Simply snipping a pair of cranial nerves can quickly render one blind or deaf. The pathways are discrete and relatively simple. On the other hand, pain is processed redundantly and diffusely, and there is no surgical procedure to relieve pain, much to the frustration of patients and doctors everywhere. Instead, pain recruits overlapping pathways and circuits throughout the body and brain that, and along with the wide-ranging neurochemistry of endogenous opioids and anti-opiates, hints at the critical role pain plays in our survival. Let's explore the vital role pain and conduct a thought experiment that can help us understand the role of endogenous opioids in our internal hardwired management of pain. Envision returning to your home and finding an intruder with whom you have an interaction that leaves you with a serious, painful, bleeding wound. Now, consider a couple possible outcomes. One is you are overcome with pain and fear and writhe on the floor until you bleed to death. This biological response is unlikely to help you survive, or, from the biological lens, it is unlikely to help you reproduce your DNA. Your DNA potentially does not survive, and you may not yet have contributed sufficiently to the survival of your species. Instead, as a result of the encounter, your brain quickly directs the synthesis of a flood of endorphins throughout your CNS, which result in blocking pain transmission, decreasing panic, therefore enabling possible escape. In this scenario, by not being overcome by fear and pain, you manage an escape. Now you find yourself safely outside. That flood of endorphins saved you. Once you are out of acute danger, it is now beneficial for you to perceive the pain. Not feeling the pain might lead you to not seek medical care, putting you at risk of bleeding to death or developing an infection. That synthesis of endorphin flood ceases. The endorphins begin to degrade, and to speed up the process of perceiving the pain, your brain now directs the release of anti-opiates to sharpen your perception of the pain in hopes you will address your wound. How do we get to this opioid crisis? There is a lot of finger pointing and lawsuits going on, but consider in 2012, 255 million prescriptions were written for opioids. That results in a lot of folks being exposed with wide-ranging individual experiences. With the use of exogenous opioids, as with any exogenous substance that alters function, our bodies and brains immediately begin making alterations that result in adaptations designed to attempt to return us to set point. Adaptations that underlie opioid addiction begin with the very first use and gain strength through repeated exposure. As a result, the dose that initially worked well no longer suffices, resulting in the need for increasing doses with resulting mounting adaptations your body is undertaking to maintain homeostasis. Opioids decrease neural activity wherever there is an opioid receptor. Now we understand how broadly opioid receptors are distributed throughout our bodies. It stands to reason then that the effects of exogenous opioids are just as broad. Some of these effects are analgesia, respiratory depression, relaxation, sleep, decreased blood pressure, euphoria, and many more. We now understand compensatory adaptations are taking place at every point of effect. The strength of these adaptations increases with continued use. Without the drug on board, these adaptations are revealed, resulting in withdrawal symptoms that are the opposite, such as pain, panting, yawning, restlessness, insomnia, increased blood pressure, irritability, and dysphoria. The longer the use, the more severe the resulting opponent process, aka withdrawal symptoms. A person struggling with opioid use disorder faces a two-fisted misery, that of the withdrawal symptoms coupled with the fear generated from their hijacked limbic system. Buprenorphine can be an important tool toward reducing harm, supporting sobriety, and ultimately assisting overcoming one's opioid use disorder. In summary, having supersaturated the U.S. population with opioids, it is logical for us to realize that with genetic variation, we will see more extreme responses to them. Many of us take an opioid and feel pain relief, but also experience a mild unease. We will discontinue the med as quickly as the pain is manageable. But there are also those whose brain upon the first use responds with euphoria. That latter group may not go on to develop an opioid use disorder, but it does increase their chances of increased and prolonged use with the scientifically predictable outcome. In the mid-90s, a THC receptor was identified and cloned. Finding a receptor is relatively easy. You tag the drug, in this case THC, with a radioactive tag. It was through this process scientists found THC was truly remarkable. THC interacted with receptors all over the brain, on the cortex as well as subcortical regions. In some regions of the brain, THC's activity was seen at every single connection. This means within our brains there is a wide and dense distribution of cannabinoid receptors, with activation of these receptors having generalized effects due to its influencing various transmissions all over our brain. It stands to reason we would not have all these receptors without some type of neurotransmitter. So then the hunt was on for the neurotransmitter of our endocannabinoid system. 2 of the endogenous cannabinoids were isolated and named anandamide and 2-AG. The endogenous cannabinoid system is atypical. These neurotransmitters diffuse upstream across the synapse. It conveys information from the receiving neuron to the sending neuron. Once these cannabinoid neurotransmitters reach their target, they alter the signal-to-noise ratio. The result is the signal is deemed important and to be given heed. Let's imagine you are early man out foraging for food and you happen across some particularly sweet and tasty strawberries. The sugar in those strawberries is essential to your survival. Millions of receptors are involved in this finding, from the processing of your senses, stimulating movement, coding of the memories to enable finding that strawberry patch again, to communicating the good news to your people. It is likely that all these connections are releasing endogenous cannabinoids. Doing so to indicate the importance of the discovery and distinguishing it from the other events of the forage that did not yield special results. Having the cannabinoid receptors located throughout the brain increases the chance that you and your people will find that strawberry patch again. What follows then should be an easy mental jump. When one ingests THC or exogenous cannabinoids, stimuli becomes marked as important and profound. Ordinary sights, sounds, and taste now seem extraordinary. With chronic ingestion of exogenous cannabinoids, there is a predictable downregulation of the cannabinoid system, making the world without THC increasingly boring and inconsequential. For the THC state-legal THC consumers, the word of the wise is infrequent and intermittent use will minimize the predictable system downregulation. An important note here. Its consumption can be devastating to the developing adolescent brain and should be absolutely avoided. We are spending very little time on stimulants, partly for time, but also because there is no pharmacological treatment. Stimulants increase general arousal and alertness, and there are few contexts in which being alert and active is contraindicated. As a result, they are widely used both legally and illegally. Cocaine and amphetamines have similar mechanisms of action. Unlike other drugs we've discussed, this group of drugs' mechanisms of action does not involve acting on a receptor. Instead, these compounds interfere with the recycling of monoamine neurotransmitters. The reduction of recycling results in higher levels of these neurotransmitters. The change of dopamine's level indicates to our brain that this stimulus is noteworthy. Amphetamines, including methamphetamine, are frequently mixed with opioids. Many folks find they are using them upon their first drug screen. As we now understand, dopamine levels are a measure of addictability of a substance. From this slide, it is easy to see the devastatingly addictive nature of meth. All I can tell you is I have no intentions of ever using a substance that is 6 to 7 times more addictive than a perfect eclair topped with a tasty chocolate hazelnut spread. There is a wide variety and potency of substances that alter perception. We are taking a focused and briefest of looks into this class. Psychedelics are in the hallucinogenic class that trigger non-ordinary states of consciousness via serotonin receptor agonism. Though this class of drugs is highly regulated, they do not activate the mesolimbic pathway. Some would therefore say they are not addictive. Interesting studies are happening with some drugs within this group of compounds, bringing cautious hope to the behavioral health community with the possibility of decreasing suffering in select disorders. Do not go out and start licking toads, but do stay tuned. There may be interesting things to learn and discover about some of these drugs. Pharmacists can significantly contribute toward reducing harm and facilitating wellness for those suffering from some substance use disorders. There are scientific, evidence-based therapies, including buprenorphine and naltrexone, that can be essential tools toward wellness. But before we can fully participate in that healing, we have to accept the disease model of a substance use disorder in both our head and our heart. We'd not withhold metformin in the person who had eaten their way into type 2 diabetes and continued to struggle with healthy dietary choices. Taking the step of separating the human suffering from the disease of substance use disorder from the behavioral manifestations may mean we have to transcend our own biological response. We come to recognize our own judgments and stigmas, own them, and overcome them. Our call to action is, in the course of therapeutic encounters with persons with substance use disorder, recognizing the space between the stimulus of the person before us, considering our response, and letting it be informed by the science of our ever-expanding understanding of this deadly disease. Thanks for your time. I hope you found this useful.